Hello viewers. Today let us study about complement system. This is Dr. P. Mary Anupama, faculty from the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam. Coming to introduction to complement system. Paul Ehrlich, he has coined the term complement. Coming to definition, it is the activity of the blood serum that completes the action of antibody. It was first discovered by Baudet in the year 1895 and he named it as Lectin. Complement system, it includes a group of thermolabile enzymatic proteins. Basic functions of complement proteins. These are involved in lysis of cell, that is lysis of bacteria or virally infected cells. Next is they promote opsonization of a particular antigen. When a bacterium is coated with the complement proteins, these are phagocytized and this is called as opsonization. Third is they play a role in activation of inflammatory responses. So some cells, they have these receptors for complement proteins and when they bind, they contribute to extravasation of the cells. And one more thing is degranulation of the basophils or mast cells can take place, which also contributes to extravasation of cells, that is movement of cells out of the blood vessels. Last one is clearance of immune complexes. When antigen antibody complexes are formed, definitely complement system gets activated and it binds to the antigen antibody complex. Some cells, phagocytes, they have receptors for this complement and thereby they phagocytize and thereby clearing the immune complexes. So these are the four basic functions of a complement system. Complement system, it comprises of glycoproteins and these are synthesized by the liver hepatocytes, monocytes, macrophages and epithelial cells of gastrointestinal tract and urinogenital tract. They are seen in the serum beta globulin fraction. They exist as inactive forms and they get activated by losing a small fragment by proteolytic activity. The larger fragment which is left out now becomes enzymatically active form. Here we can see a complement protein. It is cleaved, resulting in a larger fragment and a smaller fragment. The larger fragment, it is enzymatically active and this can bind to the cell surfaces. Say for example, as in the case of C3. C3 is cleaved into C3b, the larger fragment, and C3a, the smaller fragment. This smaller fragment, it drifts away and it is involved in inflammatory responses while the C3b binds to the cell surface. There are other B fragments which have an active role and they become enzymatically active. These are thermolabile and they denature at 56 degrees centigrade. Over 30 different complementary proteins were identified and these proteins, they are designated by the alphabet capital C, which is followed by a number based upon the order of discovery like C1, C2, C3, etc. And the larger fragment produced, it binds to the cell surface near the site of activation, while the smaller fragment, it drifts away and is involved in inflammatory responses. The complement proteins, they interact with each other during the enzymatic cascade and they form active complexes. So the order of activity of these complementary proteins, it goes like this: C1q, then C1r, C1s, C4, C2, C3, C5, C6, C7, C8 and C9. So 1 C1Q, C1R, C1S, then C4, 2, 3, 5, 6, 7, 8, 9. Others include protein B, D, P, H and I. And when you have this immune complexes, active complexes formed, we put a bar like this. Say for example C4b comes up with C2A to form C4b2A complex, which is enzymatically active. C3b joins the BB fragment, which is enzymatically active. Pathways of complement activation. There are three pathways which we'll be studying: the classical pathway, alternative pathway and lectin pathway. Classical pathway of complement fixation. This begins with the formation of antigen-antibody complex, and the antibodies which participate in this classical pathway of complement fixation are IgM, IgG subclasses G1 and G3. As such, when we see IgG, it is a monomer, where IgM, it's a macroglobulin which is planar, and hence complement fixation site is not available here, while IgG directly has a complement fixation site. This is planar initially, but when this binds to the antigen or bacterial surface, its conformation changes, and the complement fixation sites at the Fc regions are now available for the binding of complement proteins. Thus, the complement fixation is initiated. The complement fixation is initiated first by the C1 component of the complement system. This C1, it comprises of C1Q, C1R and C1S. This C1Q, it is like this. It is made up of a collagen-like triple helical structure and it has heads. These heads, they bind to the CH2 domain of antibody. So C1Q, it has 6 binding sites for the antibody. It is a very large macromolecule. The C1Q, it consists of the stalk and the head. Stalk and the head. Each in turn has a typical triple helical structure like that of the collagen, and it has the site for binding to the antibody. Once this binds, conformational changes are induced in C1R followed by C1S. This is our C1 component. The heads interact with the CH2 domain of the antibody. This induces changes in the C1R and C1S. There are two C1R molecules and two C1S molecules. These get activated. So we put a bar on Cr2 and S2. We put a bar indicating that they are activated, and these in turn activate the C4 component. The C4 component is lysed by C1Q R2S2 component, resulting in C4A and C4B. C4A drifts away and we have C4B. Next, this also activates C2 component. This C2 is again lysed into C2B and C2A. C2B drifts away while the smaller component C2A, this is just the reverse case, only one reverse case. Generally we have B larger component retaining, but here the smaller component is retained, and what you have is C4B2A complex. We have put a bar indicating that this is enzymatically active, and this we call it as the C3 convertase. This C3 convertase now splits up C3, and C3A, the smaller component, drifts away while the 3B component comes and binds here. This becomes the C4B2A 3B complex, which we call it as the C5 convertase. This C5 convertase acts upon C5 and lyses it into C5A and C5B, and C5b is now ready to bind to the membrane. All the pathways that we are studying, they usually come and merge here. So I'm stopping it here and we shall see the next pathway. The next pathway is the alternative pathway. This is independent of antigen antibody complex, and the proteins that are involved in this pathway are C3, factor B, factor D and propyridine. It is initiated by cells components of this gram positive and gram negative bacteria. The alternative pathway is mainly due to C3, and C3, it has a thioester linkage between the C3 and C3b components, and it is easily cleavable. So what do you have is C3 and C3b formation. This C3b, it goes and binds to the foreign antigen surfaces, and it can also bind to our cells also, but our cells, they have sialic acid which inactivate the C3b. The C3b, which is bound to the antigen, it exposes sites for factor B to bind. Now this factor B, which is bound, it is cleaved by factor D. So we have C3b and factor B which is cleaved, resulting in C3b BB complex, and this becomes the C3 convertase. And here is our alternate pathway. C3 is initially cleaved, that is because of the weak linkage, we have C3 and C3b formation. This C3b exposes sites for factor B to come and bind, and this factor B is acted upon by factor D to result in C3b BB complex, which is the C3 convertase. This C3 convertase again acts upon factor C3, and this C3 is cleaved into C3 and C3b. But what we have here is C3b BB3b complex, which is the C5 convertase that can hydrolyze the C5 factor, resulting in C5a and C5b. So here you can see the similarities between both the pathways. In the classical pathway, C3 convertase is C4b and 2a, while here it is C3b BB. Here it is C4b 2a 3b, which is the C5 convertase, while here it is C3b BB 3b complex, which is the C5 convertase, and we have come here again to the common point of C5b formation. From here, the formation of membrane attack complex is initiated. Once the C5b is bound, this induces again changes, and this leads to the binding of C6. Then after C6 is bound on the cell membrane, C7 will come and bind, which will induce again conformational changes. Initially they will be on the outer side of the membrane, but as they form, they start undergoing some transitional changes, some conformational changes that enables them to move down. This is followed by binding of C8 component, which impregnates into the membrane, and a small pore is formed of around 10 angstroms that is embedded into the membrane of the cells. Now after this is formed, then polymerization of C9 will be taking place. Many C9 monomers come together, they polymerize, and they form a tube-like thing which again gets impregnated onto the membrane of the infected cells or the bacterium, and now the size of the pore will increase to 100 angstroms. This entire thing we call it as the membrane attack complex. This can cause disturbance in the cell osmolarity, and it can cause loss of the cell components, and finally leading to the death of the cell. So this is the summary again. Antigen antibody complex are formed. Now this initiates binding of C1. The C1 complex once bound, the C1Q, C1R, C1S are there. So C1R, C1S, they initiate activation of C2 and C4 components. So we have C2b and C4b fragments which are formed, which are the C3 convertase. Now the C3, once hydrolyzed, we get C3A and C3B. This C3B, it goes and binds to C5, and you get the C5 which is C5b, which is formed. This C5b drifts away and binds to the membrane. Now this induces the binding of C6, C7 and C8, followed by polymerization of C9, resulting in the formation of this membrane attack complex. So because of the disturbance in the osmolarity, the cell will burst. So this is the sequence. Initially C4, C2, followed by C3, and then C5 activation. Then C5b, 6, 7, 8 and 9 forming membrane attack complex. Because of the osmolarity disturbances, the cell swells and finally it dies. So this is the representation of the membrane attack complex. So we have C5b, C6, C7 and C8. These are on the outer surface, while once C8 is formed, transitional changes will take place, and the conformation of these proteins change and they get impregnated into the cell membrane. Then C9 activation will take place and it forms a pore. See how it has crossed the membrane and it has led to disturbance in the osmolarity of the membrane and the cells will lyse. We have one more pathway called as the lectin pathway. What are lectins? Lectins, these are proteins which bind to specific carbohydrate targets, like we have this mannose binding lectins, abbreviated as MBLs. These are glycoproteins and they bind to the surface of microorganisms. MBLs, these are acute phase proteins also which participate in the inflammatory responses, and these MBLs, they are like the C1q component of the classical pathway, and these are associated with serine proteases which are called as the MASP1 and MASP2, that is membrane binding lectin protein serine proteases, SPS serine proteases. These serine proteases, they are like the C1R and C1S components. MASP1 is like C1R, MASP2 is like C1S. So these mannose binding lectin proteins, they bind to the foreign surfaces, and we have MASP1 and MASP2 which activate the C4. Now this C4, as we know, it gets split into C4A and C4B. Then we also have C2 activation taking place just like in the classical pathway. So we have C4B2A formation, which is the C3 convertase. This in turn activates C3 to form C4B2A3B complex as C3A drifts away, and this is our C5 convertase. So this C5 convertase activates the C5, it leaves the C5, C5A drifts away while C5B is there which initiates, it goes and binds to the bacterial membrane or infected cell membrane and initiates membrane attack complex formation. So what are the biological consequences of complement activation? So the membrane attack complex, it can lyse a broad spectrum of cells. It lyses virally infected cells. It's very effective in lysing gram-negative bacteria, and it can also lyse cancerous cells. The cleavage products of complement fixation, they mediate inflammation. We have C3A, C4A and C5A, the smaller fragments. They are called as anaphylotoxins. That is, they can bind to the mast cells and basophils and induce their degranulation. C5A, it induces increased extravasation of monocytes and neutrophils. C3b and C4b binding, it facilitates opsonization. That is, C3b binding after complement activation, this promotes phagocytosis. phagocytosis of this antigen antibody complex. And as we have seen, the complement, it neutralizes virally infected cells. It can result in destruction of these virally infected cells, and it is involved in clearance of immune complexes. Thank you for watching this video.